Okay, so today we're going to discuss gene therapy. So, gene therapy is a process whereby you try to correct for the problem in your genes, like the gene here undergoing mutation, and because of that, once the genes undergoing mutation here try to produce an abnormal product, and it might result here to our genetic disorders. So when speak about gene therapy, it's a process whereby you try to correct for the mutation in order for you to treat your different genetic disorders. Okay, so we have here the different method of genetic therapy or the gene therapy. The first one, since we have here an abnormal mutated genes, so you can insert a normal genes into your abnormal genes in order for that then to be corrected. The second one, you replace. The first one, you don't need to get that one, but just need to insert a normal gene in order for that then to undergo a normal process in order for that to be corrected. The second one is you just need to be replacement, so you just need to get that abnormal gene and replace that one. So wala ka nang magagawa but to replace it. Dito sa number one, pwede mo siya papanggawan ng paraan, you just need to insert your normal or your therapeutic gene, the normal gene, in order for that then to be corrected or to lessen here the manifestation of your genetic disease. Second one, so you don't have any choice here but to replace your normal gene with your abnormal gene with your normal gene. Third one, you could also repair that one by selective reverse mutation. Selective, why selective? Because again here, for sinabing selective, you are only correcting here for what is being affected in the gene. So you'll not be reversing the entire gene makeup of the person here, but rather just need to correct only for the affected or the gene in particular that has been mutated or has become a problem. In the fourth one, you could change also here, you could correct that one by changing the regulation of your gene repair. So you could have that different method, you could have drugs, treatment, or whatever it is. Okay, then we have here, so since when you are correcting for the abnormal gene, so all you need to do is to deliver your normal gene. Again, your normal gene, you call it one as your therapeutic gene that would try to correct here for the abnormal gene. So we deliver here our therapeutic gene by either having a vector or an agent of the transfer of your therapeutic gene. So the vector could be a virus. Most likely that one is your deactivated virus. So there are different viruses listed in your PowerPoint. You could have your adenovirus, you could have your different viruses. So these are here the vector. So siya magkikarry ng therapeutic gene in order for that to be inserted to the gene of the host or the patient. Another one, you could deliver also your therapeutic gene here by the use of your somatic, by the use of your stem cells. So stem cell in our hematology, so those are the cells which have the capacity to grow, to differentiate, and divide. So most likely these are undifferentiated cell or young cells. So even your white blood cells, so meron silang the normal process maturation, we have here the young cells of that, and it could grow, multiply, and differentiate and become mature. Same through with your stem cells. So all you need to do is just manipulate your stem cells. So bibigyan mo pa rin siya or lalagyan mo pa rin siya ng therapeutic genes in order for that then to in order for that to carry here the normal gene. So the somatic your stem cells once it has been already inserted with your therapeutic gene, your stem cells multiply, grow, and differentiate, then magiging normal na siya. You're able to replace here your abnormal genes or abnormal cells in your body. Okay, so those are the way of delivering here your therapeutic genes. Okay, then we have here the types of your gene therapy. So we classify that one as either your somatic gene therapy or could also your germline. Somatic gene therapy is a type of your gene therapy where it involves here your different cells in your body except your sex cells. It does not involve your egg cell or your sperm cells. Your germline gene therapy, on the other hand, involves here yours your sex cells. So use here for the treatment of your egg cell or your sperm cells. So we have here the advantages, the main advantage of your somatic cells here. So this is, we have here the main difference between your somatic cell and your germline stem cell gene therapy. Your somatic gene therapy here, so this is a method where you try to, a process whereby you try to treat your particular disease involving here all the other cells of our body except your sex cells, and that is only being treated here for that present generation. So if you try to treat that one, like the patient today, the child today, for example, this generation has have that genetic disorders, to just treat that one. But we are not sure here for the next generation. It might come out again the same genetic disorders. And that one, you don't have the assurance here, even if you try to treat the present generation, the next generation will still will have here the chance of being still be affected by genetic disorders. Whereas in your germ lines gene therapy, so this one is being able to a process whereby you try to treat here your egg cells, sperm cells, developing cells, and that could eventually be passed on to the next generation. So it would able to treat here your familial genetic disorders, familial family. So it would able to treat here your genetic disorders, okay, down the line, the entire family tree. Okay, then we have here, so first we discuss your somatic gene therapy. So your somatic gene therapy here, again, this one is involved here for the treatment of your body cells, except your sex cells. So it's used here for the treatment of diseases like your cystic fibrosis, your CF, your muscular dystrophy. We have also here your some cancers and some infectious agents. So we divide your somatic gene therapy here as either ex vivo gene therapy, or we have also here the in vivo. So ex vivo here is also called your in vitro. So when we speak about in vitro, you try to carry the manipulation of your genes here outside the body of the patient. So before it try to be returned back to the patient. Here naman sa in vivo gene therapy here, so you just directly manipulate the genes, okay, directly inside or within the body of the host or the patient. Okay, then we have here the first example of the first type of your somatic gene therapy. We have here your ex vivo gene therapy, also called your in vitro. So being in vitro, so you try to manipulate the defective gene to make that to correct for that, but you try to carry that one outside the body of the host. Then after manipulation that one, so you try to manipulate it by putting on therapeutic gene, and that's being carried by the vector, and after that one is being returned back to the body of the host and allowing it to grow inside the body of the host. Okay, so this is used basically here for the treatment of the specific organs in our body which are affected. Like for example, we have your bone marrow cells. We have also here your tumor infiltrating lymphocyte. We have also here your blood vessel, smooth muscle cells, including your liver cells for the treatment of your blindness, your corneal disease, including your eye infection like your glaucoma, and even we have also here your treatment of your leukemia and your multiple sclerosis. So how is your ex vivo gene therapy being performed or being done? So the first one, so since the patient would have here the defective na gene, so you need to take that defective gene outside of the body of the patient. Then you try to, in the laboratory, you try to manipulate that one by inserting that defective gene or the dysfunctional gene by the therapeutic gene. So in order for you to insert that one, you just need a carrier or a vector in the form of your virus. Okay, and after that one, you allow it to grow in the culture in the laboratory. Then try to check and try to harvest those which has transformed the cells. Pag sinabing you just need to collect here the transformed na cells, because only those cells, those are able to grow, because those na hindi nag-transform, so mean to say those are the defective na cells, or the entire process of taking up your therapeutic gene here from care by the vector is not successful. So therefore, what you need to harvest is only your transformed cells. Because mean to say by transform, so mean to say the therapeutic gene coming from your vector has already been inserted on your defect in the gene from your host and able to grow, mean to say able to take it up that therapeutic gene effectively. Then after that one, you try to return it back to the body of the host. Then inside the body of the host here, so your transform, manipulated the genes were able to produce here the desired product that would already have here a normal product being produced. And therefore able to correct here for the defects. Okay, advantages of your ex vivo here. So number one, so this is highly specific here. So it would only be able to be correct here, able to be corrected only those specific defects or organs of your body which are defective. And second one, this is immunocompatible between your host here and your recipient. So by the process of the transplantation in our immunology serology, so basically for you to have a successful na transplantation, the donor recipient should be compatible with one another. And there are many factors for their compatibility. One, dapat compatible sila sa kanilang ABO blood groups. Another one, HLA or human leukocyte antigen. So those are very important factors for you then to consider here that the success of your transplantation. Why immunocompatible here is because na because you try to take the part of the host here as the defective gene, nilagay mo sa laboratory, naglagay ka ng therapeutic gene, and nag-transform siya, so mean to say they are compatible. Okay, so pag ibigay mo yan sa host, try to return it back to the host, most likely the host will not mount a harmful immune response. Because most likely, pag hindi compatible, pag nilagay mo sa body ng host, your immune system will be activated that would eventually resulting here to the destruction of your incompatible na transplanted na tissues. So here, doon pa lang sa laboratory, since nag-transform siya, nag-grow siya, mean to say they are already compatible. Since you are that's the only one you are collecting naman, not the transform. Not the one not able to transform. Okay, disadvantage. So this is highly invasive process. Invasive in sense na una, dalawang process talaga ang sobrang traumatic ito sa patient because number one, kukuhanin mo sa katawan niya tapos ibabalik mo pa siya. So una para pagtanggal, so there's already traumatic or invasive. So pag binalik mo siya ulit, it becomes also here another invasive procedure for the patients.